Hi everyone, and welcome to our video on the structure of the liver. This is part of our Module 5 series, carrying on with the section on excretion. And what we're going to be doing in this particular video is focusing in on the liver. So we're going to be looking at specification reference 5.1.2b part 1, the structure of the mammalian liver, and 5.1.2b part 2, the examination of stained sections showing the histology of liver tissue. Now, before we delve into the actual structure of the liver in any detail, it's worth just taking a moment just to remind ourselves of some of these key functions that our liver performs for us. So one of the main ones is actually helping us to maintain our body temperature. And what we find is that the liver itself is very metabolically active. So what we're going to be able to do is therefore have contributions to maintaining our body temperature. It's involved in controlling blood glucose, and even back at GCSE, hopefully you remember it has that role to play in storing glycogen. It helps with regulating metabolism and the synthesis of many proteins and other molecules there. We can also store other bits like vitamins and iron, and it has this role in breaking down or degrading certain hormones and also in activating those different drugs and toxins that can then be excreted from the body as well. So we can see from this little summary list that it's got a diverse range of different roles, all of which very important for the functioning of a mammal. Let's start then with some of the basics. If we think about the liver itself, then it has blood supplied from two sources. So its first source is what we call the hepatic artery, and this is going to supply approximately 30% of the total blood supply for the liver. And secondly, it's got the hepatic portal vein, and that's going to be the other 70% roughly of the blood supply. Now, before we go further than this, it is worth remembering that when we're looking at some of our words, the word hepatic is just referring to the liver. So hepatic artery is the artery of the liver basically, and the hepatic portal vein is obviously a vein associated with the liver as well. Now if we look at the little diagram underneath, we can see we've got the hepatic artery in the red there, and then that light blue colour just coming up near the green tube at the bottom there, that's the hepatic portal vein. So what we actually have is the hepatic artery is the oxygen rich blood, so that's the stuff that's going to bring the oxygen required for all these metabolic processes. And the hepatic portal vein, notice the green bit there in our diagram, is the gastrointestinal tract, and therefore that's going to be bringing all of those bits like the glucose etc. from our digestive system to the liver itself. Now the hepatic artery, as we've mentioned, is going to bring this oxygenated blood from the heart to the liver. And our liver does need a very good supply of oxygen because it has this high metabolic activity. Now, any of these parts of the body that have a high metabolic activity are going to be requiring large quantities of ATP. And ATP is going to be made predominantly through aerobic respiration. And hopefully we remember that if it's aerobic respiration, that means we've got to have a plentiful supply of oxygen in order to fully oxidize whatever our respiratory substrate may be. Let's now consider the structure of the organ itself. And if we look at a liver, then what we have are actually two sections to it, and we refer to these sections as lobes. So we divide it up into the left lobe and the right lobe. So I've given you a little picture down here of a person just to show you where the liver sits. So you can see it sitting just beneath, obviously, that kind of bit of your ribs. So if you kind of poke around, you'll find your rib cage, and your liver basically sits just running along the top there. And you can see on your right hand side, you've got your right lobe, that's the larger of the two, and then moving towards obviously the left side of your body is the left lobe, the smaller of the two. Now, when we actually consider what happens to those lobes, they are actually subdivided into these smaller structures we refer to as lobules. And these are pretty much cylindrical in shape. And what we've got at the bottom is the diagram of a typical liver lobule. Now what we can see is in the very centre there we've got the central vein, and then if you look around the outside in this little region, what we have is basically a little collection of 3 tubes to all intents and purposes. So the blue one in the middle there of our little triple collection is going to be the portal vein. The red one is going to be our hepatic artery, and the little green rimmed one is the bile duct. So what we actually have then are these little triads, these collections of 3 tubes basically, that are then going to be associated around the edge, as we can see, and that central vein in the middle of the lobule. As we've said previously, we've got these 2 major blood vessels that bring the blood to the liver itself, hepatic artery and hepatic portal vein. What we're going to find is those 2 large vessels are then going to split into lots of smaller vessels, and that's what we see in those triads that then run parallel to those lobules. And we refer to those as the interlobular vessels. So I've given you a different little version of the diagram here where we can see we've got our individual lobules, there's 2 in that little diagram, and then around the edges we can see we've got those little triads. So we've got our bile duct in the greenish colour there, the hepatic artery in the red again, and the blue is going to be the hepatic portal vein. So we've got those associated with those lobules running parallel throughout the structure there. On the far right, we've actually got a microscope image of that same structure. So again, we need to be familiar with looking at these microscope images. Now what we see, this is from a light microscope, and the reason that we can tell this is from a light microscope is that we can see obviously whole structures here. We can see an entire lobule. In fact, we can see several lobules on here, which wouldn't be what we would see under the much higher magnifications of things like the transmission electron microscope. Then we need to be able to identify those key parts. So obviously picking out a lobule is quite straightforward because you can just draw around the edge of a lobule. So there you go, this is going to be one of our lobules first of all. Now in the very centre we've got this little white region here. So that white region in the centre is going to be our central vein. Then what we have coming around the edges, we've got our little triads, remember. So if we have a little look, we can pull out one particular triad. If we look up at the top here, try that again. So if we look up at the top, we've got our first little triad up here. Now, remember, veins are always going to have that larger lumen. So where we've got this structure with this much larger opening in that triad, that's going to be our portal vein. We then are going to have a region, and where I've drawn it's probably not too easy to see. It's going to be down here, but we're going to focus on this little area because we can see it more clearly. So if you look at the structure there, we can see there's a much darker pink ring around the outside. That's just showing us the thicker wall of the hepatic artery. And then the other structure that we're going to find on the other side of the portal vein with the thinner wall will of course be the bile duct. And we can see those obviously scattered throughout the actual structures in the microscope image. So we've obviously got our hepatic portal vein, hepatic artery. They branch into these smaller sections that then run parallel to our little lobules. And what we're then going to find is that at intervals we get these little branches from the hepatic portal vein and the hepatic artery entering the lobules themselves. So we've obviously got the main runs which are running parallel, and then every so often they branch off and enter the lobules themselves. And what we find is that those branches of the hepatic artery bringing the oxygenated blood and the hepatic portal vein bringing that nutrient-rich blood from the digestive system are going to actually combine together in that liver to form something we refer to as a common venule. So basically those individual little tubes are now coming together, and this is then going to create these channels which we call a sinusoid. So if we have a little look at the diagram on the right here, it's a bit easier to see. So we've got our blue of the hepatic portal vein, and then we've got the red of our hepatic artery. So we can see this is our little triad because we've got the bile duct on the right there. So what we can see is there's a little branch running in to the actual lobule itself. So what we've actually got, let me pick a colour that's hopefully going to stand out nicely here. We have got, no, that doesn't stand out at all. Let's try again. There we go. So we've got one little branch coming in here from the hepatic portal vein. That's obviously bringing our nutrient-rich blood from the digestive system. We've got a little branch then coming in from the hepatic artery here, bringing the oxygen-rich blood. And we can see they're actually coming together into this structure, which we refer to as a sinusoid. So the sinusoids then are basically going to be lined with these special cells called hepatocytes. And again, if we break down that word, hepato is just meaning liver, and cytes means cell. So basically, they are the liver cells that line it. And you can see them in the diagram, almost looking like little square or cuboidal little shapes there. Now, what happens is, as that blood flows along the sinusoid, what's happening is it's being brought into close contact with those hepatocytes, the liver cells. And that's where we're getting the exchanges taking place. So the oxygen, the glucose, etc. are able to then enter our hepatocytes, and anything that we want to obviously place back into the blood can transfer into it. And what we find is, as always, where we've got exchanges taking place, there are, of course, some little adaptations to make it a bit more of an efficient process. So those hepatocytes have these structures called microvilli on their surface, which are basically like little folds on their surface membrane. And the reason they've got those is to increase surface area. So if you have a look at the little diagram that's zoomed in on the right there, we can see that we've got these little foldings moving in from the surface of those cells, which are the microvilli. Now, if we have a little look at another microscope image, what we can see again, this is obviously a greater magnification because we can only see part of a single lobule. Then what we're looking at here are some of those finer structures we've just talked about. In the centre, we still have our central veins. That big white circly bit is the central vein still. But if we then have a look, we can see there are actually these little kind of white channels. So these little white channels that we can pick out are the sinusoids themselves. So the sinusoids, remember, are where we've got that coming together of the hepatic portal vein and the hepatic artery, so that we're going to have those exchanges taking place. So let's have a quick recap on the structure that we've talked about so far. We've got our liver. The liver is divided up into two lobes, left lobe, right lobe. Those lobes are then divided up into the lobules. The lobules themselves are going to have obviously those triads associated around the outside and our central vein in the middle of them. And then when we zoom in even further, we can see that hepatic artery and the hepatic portal vein come together to form that sinusoid. The sinusoids are lined by those hepatocytes, the liver cells, which of course have an increased surface area through the presence of the microvilli, and that's where the exchanges take place before the blood ends up in that central vein region. Now, you may have noticed that floating around in those sinusoids, we've got these little kind of weird purpley cells. Now, they are what we call Kupffer cells, and they're basically a specialised macrophage that's just roaming around the sinusoid, and their role is to break down any red blood cells that have reached the end of their life. So hopefully we remember that red blood cells, one of the key adaptations they've got is they do not have a nucleus, and as a result of part of the specialization, then they have a limited lifespan. So once they're actually at the point of the end of their life, they need to be broken down, and that's where these Kupffer cells come in. They detect those older red blood cells, and they're going to break them down into their little constituents, and one of the products of this process is a substance called bilirubin. And basically that bilirubin then becomes part of the bile. It's one of the bile pigments. So just to give you an idea about how that process takes place, we've got a little flow chart, if you like, on the right here. So if we go back to the very start of this, where we've got our red blood cells, the erythrocytes, obviously when they get to the end of their life cycle, then the Kupffer cells are actually going to break down those erythrocytes into hemoglobin first of all. Now, hemoglobin is obviously the protein that's found within our erythrocytes, and we are then going to further break down the hemoglobin into those globin chains, which are obviously made of a sequence of amino acids, and the heme groups. And remember, heme contains iron. So that heme group is then going to be broken down into the iron, which gets transported back to the bone marrow, so new red blood cells or erythrocytes can be formed. And the other part of it, the bilirubin, that gets transferred to the bile through those little bile ducts before it's stored in the gallbladder, and then that's where it will remain until it's obviously going to enter parts of our digestive system. And the way that it's actually going to come together is that within our actual liver itself, we have got these things called bile canaliculi. So they're basically like little channels, the canaliculi. So bilirubin and other bile pigments and bile salts are going to move into the bile canaliculi. Those canaliculi join together to form the bile duct, and that then connects to the gallbladder. So we've got all these little processes taking place within the sinusoid, and basically as the blood finally reaches the end of the sinusoid, it drains into the intralobular vessel. And that's just a branch of our hepatic vein. That's the central vein we've seen in our diagrams. What then happens is all of those individual little branches of our hepatic vein are going to join together until eventually we are back with the hepatic vein itself, which drains the blood from the liver. So it's then taking away that blood from the organ to the other regions of the body. If we take a little moment just to talk about the hepatocytes in a touch more detail, these are cuboidal in shape. So these cells have a cuboidal shape. And we've already mentioned the fact they've got these microvilli on the surface. But what we didn't specify was they've got a difference in the number of microvilli on different surfaces. So what we find is that the face of our hepatocytes that points into the sinusoid is going to have a large number of microvilli, because that's where we're obviously having the vast majority of these exchanges taking place. On the bile canaliculi side, the opposite side, they're actually pretty sparse. There's not many of them because we're not needing them for those exchanges. We also find that hepatocytes have a dense cytoplasm with lots of the rough and smooth endoplasmic reticulum and obviously the stacks of the Golgi membranes, and they're all to do with being able to produce everything required to carry out these processes. As always, I hope you found this video useful, and don't forget to subscribe to the channel so you can see when I next upload a video, and head on over to the website where you can find a range of other resources to help you in your A-level biology studies.